pretty much the summation of all that we said on Wednesday. This is how we end up having that resting membrane potential being -70. And so if you look at what that means that there's an unequal or uneven distribution of charge across the membrane. So yes, while both sodium and potassium are positive ions, so they're both cations, they are unevenly distributed across the membrane, in that there's more potassium on the outside, all right? There's more sodium on the outside, more potassium on the inside, and that is being maintained by two factors. The first factor we said was the sodium-potassium pump, and the sodium-potassium pump is pumping sodium out of the cell and potassium into the cell. And so the second factor were the leak channels, so the ion channels that allowed potassium to leak out of the membrane and then allowed sodium to leak into the membrane. Now, we talked about the difference in permeability. Remember, we said that there were more potassium channels, so the membrane potential at rest sort of more closely resembled the equilibrium potential for potassium. And so this means that there's more positive charge leaving the cell, right? If there are more doors, so to speak, or more channels for potassium, more of that potassium can leave the cell, less of that sodium can come in. So that is one reason for the fact that we have more of a negative environment left behind on the inside when the cell is at rest. All right, just the permeability difference between these two ions. So although they are both positive, they are at different distributions across the membrane. All right. The second question, how do all of the different channels get put together, and knowing how many there should be on the membrane? Okay, so you don't have to necessarily memorize how many channels are on the membrane. That's never something I'm going to ask. But I do want you to know the different proportions. I want you to know that there are more potassium channels on a membrane. And what we have been speaking about so far is a neuron, right? This is one of the examples of an excitable cell that has a resting membrane potential at -70 and has the ability to be excited. It has the ability to open up additional channels that we have not talked about yet. These are not going to be leak channels that are always open. These are going to be gated channels. So if you think back to our conversation about channels, we talked about some being leak channels that allow free flow of ions, some being gated that are only open under certain circumstances or conditions. And again, I don't want you to ever know how many necessarily are on the membrane, but I do want you to know the relative proportions. And they are all acting under different conditions. So you will have to know how a leak channel differs from a gated channel, from the sodium-potassium pump, and the mechanism by which ions can move through each of those examples. A third question is, how does alcohol affect the resting membrane potential? So this is a really good question. Alcohol does not necessarily influence the membrane potential directly, but it can change the synapse. The synapse is what we will speak about when we look at chapter 8, so that's coming up next week. And it's basically how neurons speak to each other, how one neuron can release signals, and that can be detected on the cell body of the adjacent neuron, and that's called a synapse or a synaptic junction. The communication at that area can be affected by alcohol, okay? And we'll point that out again when we talk about that next week. Another question here, are there variables that can change the way the membrane potential works? So yes, there are certain medications that we will bring up, certain toxins. For example, there's a TEA toxin that's released from pufferfish. I don't know if you ever heard of pufferfish, but they're deadly for this reason. When certain animals eat these types of fish, they release this toxin, and that basically delays the opening up of sodium channels. And so that can really influence or hamper the resting membrane potential, but also the ability to bring about an action potential as well. And we'll talk about these toxins and certain compounds and chemicals and how they influence the membrane potential coming up. Okay, so that was all the questions that I had. Any other questions, as usual, feel free to, drop them in the chat as we go. But I'm going to keep moving on here and talk about this graph, which is at the end of that last packet, and then we're going to move on into the action potential. So this graph shows us really the big picture of what we have been discussing. So we've talked about this value here. We've talked about this -70. We've talked about how we established that. Mainly the way that sodium moves, potassium moves, and how the combined movement of those two ions, along with the sodium-potassium pump and the relative leak channels, brings us to a steady state in a resting neuron of -70. Okay, we even went on to say that if you were to stick a voltmeter into a resting neuron, that's the reading that you would get, -70, which means there's more negative charge inside the cell relative to the external environment. Now, we can think about this as sort of our starting point, our base point, our default in a resting neuron. What this allows us to do is be able to change this value and bring about what's called an action potential in this neuron. Okay, and in order for us to do that, we now have to manipulate the channels that are open, which will further manipulate the flux of ions, right? The movement of ions, which will change our membrane potential. Okay, we're going to think about the movement of sodium. Sodium wants to get into the cell. So if you open up excess sodium ions, you're going to see a flux of sodium moving into the cell according to its ECDF, its electrochemical driving force. The opposite is true for potassium. Potassium wants to get out of the cell. And so if you open up additional potassium channels, it's going to move according to its ECDF. It's going to want to get out of the cell. So this is a way that we can change the membrane potential. We can simply open up more channels. We can open up more doors or more passageways that will create the environment where ions want to move in their normal gradient according to their ECDF, and they will change the membrane potential. Okay, so that is what we're going to speak about going forward. We're going to talk about how we can open up sodium channels. Remember, sodium's equilibrium potential is very, very way high up here. It's very positive. It's positive 60. So if you open up additional sodium channels, more sodium rushes into the cell, and the cell's membrane potential is no longer going to be -70. It's going to start increasing as it tries to approach the equilibrium potential for sodium. Same thing is true for potassium. If I open up extra potassium channels, potassium's equilibrium potential is way down here. It's -94. That's going to cause an opposite direction of movement in terms of the membrane potential. It's going to want to get way down lower than -70, down to -94, which is approaching the equilibrium potential for potassium. So the big picture that we want to say here is that we can manipulate that resting value. We can manipulate that membrane potential by simply opening up channels, additional channels that will change the membrane potential. And in doing so, we can spike the membrane potential to bring about an event that's called an action potential, which we can use to signal other cells and other neurons. All right. Okay. So let's go ahead and move on to our second packet here. I'm going to skip over the checkpoints or polling questions, and we will look at those at the very end. Okay, so let's begin with our lecture objectives here. So number one is to describe the various properties of graded potentials, including the direction of change in a potential, the magnitude of change, and temporal and spatial summation. And we're going to talk about how graded potentials in neurons can trigger what we just described, right? The action potential. And then we'll get on to the other objectives going forward. But let's start off with objective one. So a potential is basically a type of electrical signal. So we've been building this story that we can move ions in and out of the cell. But we want to remember why we move the ions. The movement of ions allows us to generate a signal. This signal is what we use to communicate. This is why the nervous system is the system of communication. Neurons can speak to each other. They can send certain signals from the brain all the way down to your fingers and toes, right? Neurons can speak to the heart. They can innervate the heart and give the heart instructions about how fast or how slow to beat. Neurons can also stimulate and communicate with muscle. So neurons have the ability via these types of signals to send instruction to all of our other organs and structures, blood vessels, the heart, muscle, etc. So the way that we send those signals is via potentials. Okay, now the bigger the potential, the more impact or the more signal it's going to have or have the ability to transmit. But if we think about the smallest type of signal, that's going to be called a graded potential. So a graded potential is a very small change in the membrane potential. It can communicate only for a short distance of time. It's not sustainable. It can't travel for long periods the way that an action potential can. And it really is just the smaller change that can accumulate together to result in a bigger change, which is the action potential. Okay, the action potential, on the other hand, is a larger signal. It's a larger potential. It has more ability to transmit over long distances and for longer periods of time, and it is a bigger magnitude in terms of the change of the membrane potential. So just to illustrate that here on our images here on the right. So here is what a graded potential looks like. Again, very small blip or small change or bump in the membrane potential. It can be a positive change or it can be a negative change. Unless a graded potential gets to negative 55, so remember our cell is starting at negative 70, which is down here. And we can change the membrane potential. We can change it to negative 60, to negative 65. Until we get to negative 55, we are not able to initiate an action potential. So negative 55 is called the threshold value. When a graded potential is strong enough to reach negative 55, then it will spike the membrane potential to become a more meaningful, larger action potential. All right, and then that will result back down into rest. Action potentials, on the other hand, are bigger changes in the membrane potential. We go from negative 55 all the way up to positive 30, and then we recover back down to rest. This type of potential has the ability to communicate for long distances and to send more meaningful signals, again, to neurons, to muscle, to other organs in the body. But in order to get to an action potential, we need to first have enough smaller graded potentials that get us to our threshold of negative 55, and then we can result in an action potential. Okay. Let's look at a drawing here. So let's illustrate what we mean by graded potentials versus action potentials. And let's look at the different areas that they are found on a neuron. All right, hang on, bear with me 1 minute. Okay, so we are looking at types of potentials. Let's first start by describing what a neuron looks like. So on a neuron, we've got the proximal portion, which is described as the cell body, sometimes called the soma. Oops. We've got the longer central portion, which is the axon. And then towards the distal or end part of the neuron, we have this bulge, which is the axon terminal. Okay, so let's start by just describing these regions or labeling these regions. So up here is our soma. Those sometimes called the cell body. Okay, this is our axon. And then this is the axon hillock, or the axon terminal, excuse me. Other important regions are the axon hillock. The axon hillock is described as the neck of the neuron. So this narrowing between the soma and the axon, this is called the axon hillock. And then we also want to think about the projections coming off of the soma, which are called the dendrites. So let's draw these in as well. And the dendrites are really the way in which other neurons can signal onto this soma, or the soma of this neuron. So these are called the dendrites. And then this is called the axon hillock. Okay. All right. And so we want to think about, let's actually put a few other neurons here so we can illustrate the region that's called a synapse. So if we think about a synapse is a cell-to-cell junction or a cell-to-cell communication. So think about another axon terminal communicating onto this neuron via this junction, right? So we have other neurons where their axon terminals, right, this structure down here is also sending signals onto this neuron, all right? That region is called a synapse. All right, so the area where one neuron speaks to the other is called a synapse, sometimes called a synaptic junction. So let's point that out as well. And so we would also have a synapse at this distal end as well. Okay, now we want to think about two different types of neurons. As we speak about these signals communicating from one neuron to another, we have a presynaptic neuron and a postsynaptic neuron. So in this example, this would be our presynaptic neuron. And then this main neuron that we're looking at more closely would be our postsynaptic neuron, right? Because it's on the other end of that synapse. So this is our postsynaptic. All right. So let's talk about the two different types of potentials that we have mentioned. Now, let's see. So someone's asking, is the neuron that's being connected getting the message? Really good question. So this is the neuron that's receiving the signal, right? This is the neuron, the main larger neuron that we're drawing, that we're looking at. This is the one that is receiving the signal or the input. This is the one that would be considered to send the signal, right? These are all sending signals here and receiving the signal. Now, because this is a relay chain of communication, the signal will be picked up by this neuron. And guess what? It's going to be relayed down the axon and transmitted to another neuron. So this neuron that we're focusing on is not only receiving input and signals, but it's also going to be sending those signals to other neurons as well, or to muscle, which can be synapsed by neurons as well. So we don't want to think about it as being only the sender or receiver, because it's actually doing both, okay? Just at different points. So it's receiving information proximally and then submitting or transferring that distally. Okay, let's talk about where we have different channels along this membrane. Okay, let's talk about where we have different channels. We have ligand-gated channels on the soma. So on the soma or cell body up here, we have what are called ligand-gated channels. Okay. Oops. All right, so these are ligand. Oh no, I don't know why my pen changed. Suddenly, hang on. Okay. So these are our ligand-gated channels only found on the soma. On the axon itself, we have voltage-gated channels. And I'll talk about what these terms mean just in a minute. So here we have our voltage-gated channels. And then on the axon terminal, we have calcium channels. Okay. The voltage-gated channels are going to be the most important ones, and we'll talk about why later on. But I do want you to know where we have these different types of channels. Now, we also have leak channels that we know are always on the membrane, right? And we have our sodium-potassium pump, which we know is also always on the membrane, right? So we're not going to draw those in, but I want you to understand that in order for our neuron to be at rest, those two channels are automatically on the membrane, the sodium-potassium pump and our leak channels that we have described before. As it pertains to the action potential, these three channels are going to be really important. How do we excite this neuron beyond that -70? Okay. Now, on that basis, we can think about two different types of potentials. And so graded potentials are going to occur along the soma or cell body, right? So this can be a cutoff point. Anything before the axon hillock, these are going to be considered graded potentials. And so the axon hillock is going to be the point where we determine whether or not we have met that negative 55. If you recall on the previous slide, we talked about negative 55 being the threshold. So the axon hillock is the center that says, has there been enough smaller graded potentials here that now puts us at negative 55 to where we can stimulate an action potential? Because beyond the axon hillock, this is where we have the action potential. All right, this is where our action potential begins. Okay, the action potential will start here at the axon hillock, and then it will propagate down the length of the axon. And when it gets to the axon terminal, it's going to stimulate the transfer to another neuron. Okay, and we'll talk about more of the details as we go along, but this is really the big picture of what we're going to talk about. So the neuron is now at rest. How do we use these smaller graded potentials to get to our threshold of negative 55? And at negative 55, we're going to spike an action potential. That's going to propagate down the length of this entire axon. It's going to reach the axon terminal, and then that chemical signal is going to be transferred to another neuron, and that process will continue again and again and again. This is what I mean by saying that a graded potential cannot go very far. A graded potential cannot propagate down the length of the axon. It's not strong enough. An action potential is. An action potential can propagate through longer lengths and for larger amounts of time because it is a more, it's a stronger signal or a stronger change in the membrane potential. All right, so here next to axon hillock, I'm just going to put in brackets negative 55 thresholds, right? This is the value that we have to get to in order to stimulate an action potential. And so the axon hillock is sort of measuring that, right? I'm going to put a question mark here. It's really measuring, are we at negative 55? If yes, we'll make an action potential. If not, we won't, right? We'll just lose that graded potential.